Hello everyone. Today we are in the last month and in the last day of this year. So wishing you all a happy morning. In our previous class we have discussed regarding the overview of cell cycle and cell division. So we had an overview of the cell cycle, how we are going to learn about it, what we are going to learn about it. We had an overview. Today, let us discuss regarding the mitosis. Mitosis is otherwise called as M phase. We can call this M phase as the most dramatic period of the cell cycle. Why is it called so? Why do we call it as the most dramatic period of the cell cycle as this M phase or mitosis? Why do we call so? Because if we talk about the early prophase, late prophase or anaphase or telophase, during this stage, the Golgi complex, endoplasmic reticulum and nucleolus will not be there in these particular stages. So we cannot see here this image. You can see here the early prophase, in the late prophase we are not able to see the endoplasmic reticulum, we are not able to see the nucleolus, we are not able to see the Golgi complex. Okay, we will not be able to see here. But during the end of the telophase stage, we can say during the telophase stage, again this Golgi complex, endoplasmic reticulum and nucleolus appears again. It appears again. So that's what we call this M phase as the most dramatic period of the cell cycle. So I have told you mitosis is otherwise known as M phase. Otherwise it is termed as the most dramatic period of the cell cycle. Now you might have understood, you might have got a clear concept about this mitosis. So why is it called so? Because during the early prophase stage or the late prophase stage, we are not able to see any of this endoplasmic reticulum, nucleolus, or we are not able to see the Golgi complex. So at the end of the telophase stage, we can find these endoplasmic reticulum, Golgi complex, nucleolus, again it will be appearing. So that's what it is called as the most dramatic period of the cell cycle. Now moving towards. So coming towards the second point, mitosis is called the equatorial division. First I have told mitosis is called the dramatic period of the cell cycle. I have explained the first point to you. Hope it is clear to you. Now the second point, mitosis is the equatorial division. Why is it called so? Because the parent cells, inside the parent cells, the number of the chromosomes and the type of the chromosome will be the same as in the daughter cell also will be the same. पेरेंट सेल में जो क्रोमोसोम नंबर जो है, टाइप जो है, उसी के जैसे ही डॉटर सेल में भी होंगी. Understood? So that will be the same. That means the inside the parent cell, the chromosome number, the chromosome type, all this you can see in the daughter cell also the same. The same number of chromosomes, the same type of chromosomes you will be able to see. That's what the mitosis or the M phase is called as equatorial division. Hope it is clear to you. The second point is clear to you. Moving towards the third point. Third point is that the cell division is a progressive process. Cell division is a progressive process. So we will not be able to draw a clear-cut lines here in between. We will not be able to draw in the, we cannot say that when is the prophase stage starting or when is the anaphase stage starting or when the telophase ends or when the metaphase comes to an end. We will not be able to draw a clear-cut lines in these stages. Understood? So, cell division we can say that it is a continuous process. It's a progressive process. So, this we have to remember. In between there is no pauses. Okay, it is a continuous process that you have to remember. So, in the overview of cell cycle, as I have told you, mitosis will be involving two steps. What are the two steps we have learned in the overview of the cell cycle? They are karyokinesis and cytokinesis. First, let us learn about the karyokinesis. When we talk about the karyokinesis, let us see the first diagram here. The first image here, that is the interphase stage. As I have told you, interphase resting stage, that is the preparatory stage. टू सक्सेसिव एम फेज के बीच के स्टेज जो है उसी को इंटरफेज कहलाते हैं। टू सक्सेसिव एम फेज के बीच के जगह जो है फेज जो है उसी को क्या बोलते हैं? इंटरफेज स्टेज बोलते हैं। So most of the cells, 95% of the duration of the cell cycle will be remaining here in the interphase stage. 95% of the cell will be remaining in this stage only, because this is the preparatory stage. This is the resting stage. Here all the preparation is taking place. Here all the complete formation is taking place. It is ready for the division. Cell is ready for the division. The complete preparation for the division. Here everything is being ready in the interphase stage. Understood? So first, when we talk about the karyokinesis, we have to talk about the interphase. Here in the first image we have, you can see the interphase stage. So in the interphase stage, as I have told you, that is the preparatory stage, that is the resting stage. So cell divide करने के लिए complete तैयारियां कर चुके हैं, division होने के लिए complete तैयारियां कर चुके हैं. That is the stage termed as interphase stage. Now moving towards the early prophase stage. So next, moving towards the early prophase stage. So this is the image of the early prophase stage. As you can see here, the aster. Aster is the very important part in this early prophase. So here, as you can see here, nuclear membrane. This disintegrates. Nuclear membrane slowly it disintegrates and the nuclear membrane disappears here in this stage. In early prophase stage, nuclear membrane disintegrates and disappears. And chromatids जो है इसमें, the chromatids that it is present here, that will become condensed, that will become entangled here in this stage. Which stage? Early prophase stage. So the chromosomes, the chromatids will be condensed and it will be entangled. And here you can see here the asters, they will be starting to move towards the different poles. So the aster will be moving towards the different various poles. See, you can see here it is moving towards the various poles. And also you can see here the spindle fiber formation. You can see here in the image the spindle fiber formation. So hope early prophase stage what is taking place you might have understood. So aster is very important here in the early prophase stage. Nuclear membrane, as you can see here, it slowly disappears. Okay, it disappears. Then it disintegrates. Chromatids becomes entangled. It becomes condensed. Aster starts to move towards the various poles. Various poles you can see here two different poles, opposite poles it is moving. Then the spindle fiber formation is taking place in the early prophase. Now moving towards the late prophase stage. So in the late prophase stage, you can see here in the third image, in the late prophase stage, what is happening? So condensation of the chromatin material is taking place in the late prophase stage. Condensation of the chromatin material is taking place in the late prophase stage. So centrioles will be moving towards, as you can see here, centrioles are appearing again. Centrioles are appearing again. It will be moving towards the opposite poles. Opposite poles में जा चुके हैं. Now chromatids and chromosomes will be visible here in this stage. We can see the chromosomes and the chromatids. We can see here it is visible here now. In the interphase stage, it was not visible. Here in this stage, it was not visible. Interphase stage, we cannot see that. It was entangled. It was condensed. So here in the late prophase stage, you can see the chromosomes, chromatids, everything is visible. Then microtubules will be assembled together. Here you can see the microtubules. It will be assembled together. Golgi complex, endoplasmic reticulum, nucleolus, everything appears in this stage. So here everything, the Golgi, as I have told you in the early stage, interphase stage, Golgi complex in the M phase, first stage it will not be there. This Golgi complex will not be there, endoplasmic reticulum will not be there. So that's what we call it as the most dramatic period of the cell cycle. Then here it appears again, Golgi complex appears, endoplasmic reticulum appears, and nucleolus, everything disappears. Okay, here it disappears. All these things disappears. That's what I told you. It is the most dramatic period of the cell cycle. But here you can see here the spindle fiber formation. spindle fiber formation is taking place and the centromere is being formed here. So, in the late prophase stage, what is happening? The chromatids as well as the chromosomes are condensed and centrioles moving towards the opposite poles. Centromeres appears. Chromosomes and chromatids are visible here. Spindle fiber formation takes place here. And the nucleolus, endoplasmic reticulum, Golgi complex, everything disappears. Okay, everything disappears. Now moving towards the metaphase. Now moving towards the metaphase. So metaphase stage is the important, most important. Metaphase में खास जो है, what is the most important thing in the metaphase is that if we wanted to study about the chromosome number or the morphology of the chromosome, everything we can study in details in the metaphase stage. Okay, we can study in the metaphase stage. Why? Because it is the best stage to learn about the morphology as well as the number of the chromosomes here in the metaphase stage. We can see here, the spindle fiber formation is also taking place here. The kinetochore. Kinetochore is the middle. You can see here the chromosomes are attached to the kinetochore. It is a disc-shaped structure. We have learned earlier also, previous classes also we have learned. So the kinetochore goes in the middle level, and all this, the chromosomes reaches in the equator level. You can see here the chromosomes reaches in the equator division or equator level. So we can see here the kinetochores also. Kinetochores are also attached here. जो prophase में शुरू हुआ है, so this started to disintegrate in the stage of prophase, and now in the metaphase stage it is coming completely to the equatorial division. So hope this metaphase stage is also clear to you all. So if you wanted, why I have told you the metaphase stage is the most important stage to study the chromosomes, morphology as well as the numbers. So it has, since it has been attached to the kinetochore and it is clearly visible here, the condensed one, the entangled one is separated and coming to the equatorial division. So hope these are all clear to you all. Interphase, the late prophase, what is happening in the late prophase, early prophase, what is happening in the late prophase and in the metaphase stage. So in the interphase stage, we have learned that it is a preparatory stage that you have to remember, resting stage. Most of the 95% of time is taking place here in the preparatory stage or in the cell cycle, and the next stage, early prophase, the nuclear membrane disintegrates and astes starts to move towards the pole in the early prophase. In the late prophase, what is happening? In the late prophase, centromere is being formed, spindle fiber is formed, and microtubules are being formed. And in the metaphase stage, that is coming to the equatorial division. So hope this mitosis M phase stage is clear to you all. Please make this diagram, learn, make a short note of it, and may God bless you all. Thank you so much.